Welcome everybody, and it's a great pleasure of mine to welcome Grant Hughes to the school. I was lucky enough to be invited to teach on a course in Rio a few weeks ago, and doubly lucky that Grant was also teaching on the same course. So that's when I had a chance to meet him and get to know his work in a bit more detail. And I'm sure there'll be something for everybody in the audience. As I think Grant will focus on nearly every pathogen that is important to us in the School of Tropical Medicine. So Grant started his PhD with the infamous Scott O'Neill in the University of Queensland. He then moved on to work in Jason Rasgon's lab at Johns Hopkins, and then moved with Jason to Penn State. And then he set up his own lab in the University of Texas Medical Branch. And so over to you, Grant.
Okay, thanks Mark, thanks for that kind introduction, and thanks for the invitation to visit. It's great to see all your fantastic facilities, and I've had lots of really enjoyable conversations with everyone here, and I'm sure that's going to continue throughout today. So I want to tell you a bit about some of my work and how we can exploit microbial symbionts to control mosquito-borne disease. So I'm going to talk about a symbiont that probably many of you are quite familiar with, and that's Wolbachia, but I'll be talking about this symbiont in Anopheles mosquitoes and how that affects the various pathogens that they can transmit, looking at how the microbiome can influence Wolbachia transmission, and lastly touch on some of my more recent work where we're talking about, or we're looking at using more generally the microbiome and the gut microbes of mosquitoes and how we can exploit them for control of different diseases. So a bit of background about Wolbachia. Perhaps many of you are already quite familiar with this, but for those that aren't, Wolbachia is an obligate intracellular symbiont, and you can see it here residing within an insect cell. It infects a diverse number of different insect species as well as nematodes, but in insects it's thought to infect around 50 to 60% of species, so it's quite ubiquitous in nature. Its success in infecting this many species has been attributed to its ability to manipulate the reproduction of its host. And it does this because Wolbachia is vertically transmitted, and it gives the infected female a fitness advantage over an uninfected female, and by this mechanism Wolbachia can sweep through an insect population. Now in the last 5 to 10 years we've become more aware that Wolbachia can block diverse pathogens in insect hosts, and so coupling the ability to sweep and spread through populations and block pathogens, it makes Wolbachia an attractive agent for controlling arthropod-borne disease, and the Eliminate Dengue group are exploiting these phenotypes and actually releasing Aedes aegypti mosquitoes into the field to control dengue, Zika and other pathogens. So I want to talk not about Aedes aegypti, but rather Anopheles mosquitoes, and as you all well know that these transmit Plasmodium parasites that cause malaria. So Wolbachia has a rather strange and peculiar association with Anopheles in that for a long time we thought that none of the mosquitoes, the Anopheles mosquitoes, none of these species were infected with Wolbachia. And so this is a list of Anopheles that were screened by PCR and all being found to be negative. Now, coupling this with the inability to make a stable line, people thought that there was that Anopheles were refractory to infection. But this has changed in the last couple of years, and we now know that there's a stable or a natural infection in Anopheles gambiae, and people have been using microbiome sequencing to identify that. And recently a stable infection was developed in Anopheles stevensii. But despite this, I still think there's this peculiarity within the Anopheles genus, if you think normally that 50% of mosquitoes should be infected. Now, I wanted to give you a little bit of methods of how we're infecting mosquitoes and how that differs from the traditional methods. Usually people, to transfer Wolbachia between species, they inject embryos. For 20 years, people have been trying that approach and weren't successful. So when we did this work, we looked at a different method, and we were purifying Wolbachia from cell lines, and you can see Wolbachia here in pink in the cell lines, and we would purify that and directly inject that into the adult mosquito. And so when we did that, the mosquito became infected. And so our idea was then we would get progeny from those injected mosquitoes and select for infected progeny and use those progeny to rear a stable line. Now, the one benefit of this approach compared to the embryo injection technique is that now we have infected adults. They're transiently infected, artificially infected, but they're infected nonetheless, and now we can use those mosquitoes that are infected to tease apart the biology between Wolbachia host associations. And so we looked at the interaction with the microbiota and how Wolbachia affects pathogens. So just to give you an idea of what the infection looks like in these transient infections, here are images, fish images, and Wolbachia is in orange here. And we see Wolbachia localising in the abdomen, in the thorax. We see Wolbachia in the head, possibly in some of the nervous tissue. We see Wolbachia in the mouth parts, and this could be Wolbachia circulating in hemocytes. Predominantly, the heaviest infection is in the fat body of the mosquito, and this is an important immune tissue. And here we see Wolbachia again circulating or in hemocytes, which are bound to a Malpighian tubule. And so looking at these images, these are quite similar to what we see in stable infections. So we thought by using this approach, it was a good tractable method to tease apart the interactions between Wolbachia and the host. So the first thing that we were interested in was looking how Wolbachia influences Plasmodium dynamics in the mosquito. And so the first parasite we looked at was Plasmodium falciparum. We screened two strains of Wolbachia here, so the WALB strain, which is from Aedes albopictus, and the WMEL pop strain, which was from Drosophila melanogaster. So we injected these mosquitoes, and then 7 days later, after injecting them with Wolbachia, we gave them an infected blood meal and then measured the oocyst intensity. And so what we saw was that Wolbachia indeed was suppressing Plasmodium falciparum in the mosquito. And so this was the first time someone had found that Wolbachia could interfere with a human parasite of malaria. So this was quite exciting and suggested that we could possibly use this approach for malaria control. We also we looked at other parasite species, and here we see that Wolbachia can actually enhance Plasmodium berghei. And so we were quite surprised by these results. This was the first time pathogen enhancement had been reported in this system. Usually Wolbachia was this panacea which would interfere with every pathogen, but perhaps that is not the case. And so here we're seeing enhancement by the ALB strain. And again, we've done some more work in other rodent systems, and this is Plasmodium uellii, and again we see enhancement. In this case, it's by both strains of Wolbachia. And so it could be the case that there's complex interactions, and the phenotype against the pathogen is dependent on the Wolbachia strain and the host. Now, it appeared that a few more papers came out, and it appeared that Plasmodium parasites might be particularly susceptible to this pathogen enhancement phenotype. And so we reviewed the recent literature and looked at the effects of Wolbachia in different hosts against Plasmodium. And so here is the work that I just talked about, where we see enhancement in these rodent models. In some naturally infected mosquitoes, we see that Wolbachia can also enhance some of these avian malaria parasites. Importantly, from a disease control perspective, all the studies with falciparum are finding that Wolbachia is suppressing the parasite. Now, the question is, what's going to happen in these other human parasites? And the answer is we just don't know at the moment. No one's looked at that. But I think that's really important, because there's cases where Vivax and falciparum are co-circulating, and so you wouldn't want to develop a strategy to interfere with falciparum, and that could inadvertently enhance another parasite. So I think this is something that really needs to be evaluated before any of these strategies can be implemented. Now, to shift gears from malaria to a virus, we knew that Anopheles transmitted on Yong-Yong virus, and so it's the only virus that this mosquito or these mosquitoes vector. And so we wanted to firstly just look at the interactions with Wolbachia, but particularly given that we knew that in Anopheles we're seeing this enhancement phenotype, and so we wanted to know what would happen with Onyongyong. So a bit of background about Onyongyong. It's an alpha virus closely related to chick. It was isolated in the late 50s. It causes the classical symptoms in humans that these arboviruses do: rash, fever, arthritis. There's been a few outbreaks, nothing too severe, but again, we could have said for Zika, we could have said that a couple of years ago. And yeah, it's the only virus that these mosquitoes are transmitting. So to gauge the interaction of Wolbachia and Onyongyong, we did some experiments in cell lines, and that's another reason why this is a nice system to work with. And now we can tease apart these interactions in vitro, where you can't really do that with Plasmodium. And so we had two cell lines infected with Wolbachia. They were the WMEL and the ALB, and they're in this SWA5B, which is an Anopheles cell line. And so what we see when we challenge with virus, that these Wolbachia strains really significantly interfere with virus. So that was encouraging. We then wanted to demonstrate or identify what's happening in the mosquito. So in experiments similar to the Plasmodium work that I described before, we injected these Anopheles mosquitoes, and we're doing this with Gambi and Stephen's eye. So we injected them with Wolbachia and had an injected control line as well, and then challenged these mosquitoes with an Onyongyong virus infected blood meal. And so again, here we're seeing in the mosquito that both strains of Wolbachia are suppressing viral titer in the mosquito. Now, as I mentioned, we had to inject these mosquitoes. They weren't stably or naturally infected, and Wolbachia wasn't being maternally transmitted like it does in nature. And so we wanted to address how the virus would interact in a stable infection. And so what we did was use Drosophila melanogaster, which is naturally infected with Wolbachia. And we have lines here that with two strains, either the WMEL-CS or the WMEL line. And we had a line of flies that we'd cleared of their Wolbachia infection. And so in this case, we injected the virus into the fly. Obviously, you can't give a fly an infected blood meal. But again, in this stable association, here we're seeing that Wolbachia can suppress the virus. So we were confident from these data, plus that of the in vitro work, we were confident that Wolbachia could inhibit the virus. And so we had a really nice tractable system to tease apart these interactions, and so we wanted to do some further experiments on the mechanism behind this pathogen interference phenotype. And so there are a couple of theories about how Wolbachia blocks. The first is that when you transfer Wolbachia into a new host, you can get this upregulation of insect immunity, and that is the mechanism behind interference. And so there's evidence for this in transfected mosquitoes. RNA-seq and microarrays show that the immune pathways are upregulated. But conversely, flies that are naturally infected, if their Wolbachia is introgressed into mutant fly lines which have these pathways ablated, they can still protect. And so in this case, immunity doesn't seem to be important, so it could be something else. Another theory is that Wolbachia is sequestering essential nutrients, and so the main nutrient people have been investigating is cholesterol. Again, in a fly system, when flies are fed on high cholesterol diets, these flies, Wolbachia can't protect in these flies, and so when there's an abundance of cholesterol, the virus proliferates, and so that suggests that cholesterol is this limiting nutrient. So we wanted to investigate these two theories. Now, at the time when we were doing this work, a paper came out from the Christofides group that looked at the immune response of Anopheles mosquitoes to Onyongyong virus. And what they found was that there were a variety of different immune genes were upregulated, and when they knocked these genes down, and this is the data that you're looking at here, when they knocked these various genes down, the virus titer increased in the mosquito, suggesting that these immune genes were antagonistic to the virus. we were wondering, was Wolbachia upregulating these genes, and that was the mechanism behind interference. And so we did some qPCR on these select genes in insect cell lines, and we found two genes, this lysozyme 4, which is an antimicrobial peptide, and ML1 protein, which is a putative recognition receptor. And Wolbachia increased the expression of these genes in cell lines, and both strains that we were working with had this same increase in expression. So we thought this could be the possible mechanism behind the phenotype. Now, just to give you a quick refresher on insect immunity, there's a couple of major pathways, the IMD and the TOL pathway. IMD is thought to control bacterial infections as well as other things. It has some anti-plasmodium phenotypes. TOL is an antifungal pathway but also has antiviral properties. So where do our genes lie in this pathway? Well, so lysozyme is an effector molecule, so it's downstream in the pathway. That ML1 is annotated on the mammalian TOL pathway, but that's why I've got both mammals and insects here. There's no annotation of that, but based on our work and from Christofides' work, we think that there's ML1 is binding with TOL here. So that's more upstream in the pathway. Also, another gene I want you to become familiar with is cactus, and so cactus is the negative regulator of the TOL pathway. So when cactus is upregulated, the pathway is turned off. Conversely, if you use RNAi to silence cactus, you can turn the pathway on. So by using RNAi, we can up or down regulate these pathways. So that's where our genes of interest lie. So we were interested in the TOL pathway. Now, to gauge how, if Wolbachia was co-opting these pathways, we did this experiment by, again, injecting mosquitoes with Wolbachia, but we also co-injected with RNAi. And so if you look on the left-hand side here, and so this was done in both Anopheles gambiae and Anopheles stevensii. So if you look on the left-hand side here in these uninfected mosquitoes and uninfected by Wolbachia, when we knocked these genes down, we increased virus titers. And so this was basically recapitulating the work of the Christofides group. But when we have Wolbachia in the system, so firstly we see that the viral titers are lower because Wolbachia is suppressing the virus. But again, when we're knocking down these genes, the level of Onyongyong virus increases. And so what we were interested in was this change in expression. Because Wolbachia was lower, it's hard to compare these, but when we looked at the relative change or the percentage increase in virus titer, we see that it's a lot higher in the Wolbachia infected background. And so this was pointing to us that this TOL pathway was important in protection. So we then went on to do some qPCR and look at gene expression on select immune genes, and we looked in a Wolbachia infected and uninfected background, as well as viral infection and uninfected. So we're looking at the interaction here. If you just look at the Wolbachia uninfected, you can see that when you add Onyongyong virus into the system, it's suppressing the TOL. Where by comparing the blue columns, when Wolbachia is infecting the cells, Wolbachia is elevating TOL. But also we have this interaction where when both Wolbachia and Onyongyong virus are present in the cell, the TOL pathway or the TOL gene is highly upregulated. Now we looked at the various other genes, and you'll notice that the absolute numbers might change, but the pattern is the same. So for ML1 and for lysozyme 4. When we look at IMD, we don't see that pattern. And again, this was suggesting to us that TOL was important and that there's this interaction between Wolbachia and the pathogen. To further validate the TOL pathway was important in these interactions, we did some RNAi experiments, and so we're knocking down these various genes, and that's in these experiments all in a Wolbachia infected background, so cells infected with the WALB strain. And so we have our control here, our LAC-Z control, and we've challenged these cells with Onyongyong virus, and that's our output. And so this is normalized to 1. This is our control here. When we knock down IMD, we don't see any change in virus titer. Conversely, when we knock down the ML1 gene, virus titer increases, so now Wolbachia can't protect against the virus. Similarly, with when we knock down TLR4, we're seeing an increase in virus titer, and when we knock down both of these genes together, we see this additive or synergistic effect, and so now a virus is really replicating quite efficiently in the cells. Now, the next two columns, what we're doing is we're knocking down the toll pathway upstream, but then we're turning it back on by knocking down cactus further downstream. So we're knocking it off and then we're knocking it back on. And so when we do this and now the pathway is functional again, we see that Wolbachia can indeed block against virus. So for us, this was suggesting that Wolbachia was very important in the, the toll pathway was very important. Wolbachia was hijacking the toll pathway, and this was the mechanism, or a mechanism for interference. Now, I mentioned another hypothesis was cholesterol, and that potentially there's this resource competition between cholesterol, or between Wolbachia and the pathogen for cholesterol. So to gauge if this was important in our system, again, we're exploiting the tractable in vitro system and cell line system. And we spiked the cells with varying concentrations of cholesterol. Here, these are the Wolbachia uninfected cells, and so we've challenged again with virus. And what we see when we increase cholesterol, we don't see a change in virus titer in the uninfected background. When Wolbachia is in this system, the first thing to point out is when there's no cholesterol, Wolbachia is blocking against the virus, but as cholesterol concentration increases, Wolbachia, the virus increases as well, and it goes well above the levels of the control. Now, to us, this didn't seem like there was a resource competition. If we were to see resource competition, we would probably expect that we would get some increases here in virus in the control line, and we probably wouldn't expect viral titers to go above the control line here. So we thought something else might be occurring. So we delved into the literature and did a bit of reading on the interaction of cholesterol. And what we found was that there's this interaction between immunity in some mammalian systems. And so there's evidence that in invertebrates, cholesterol can activate TLR4s, which are in the toll pathway. These TLR4s accumulate in cholesterol-rich endosomes, and that these interactions are important for various diseases. And cholesterol can also induce reactive oxygen species, and we know that ROS can actually turn on the toll pathway in Aedes aegypti. And so there was this link between cholesterol and immunity, which we wanted to investigate further. So to do that, again, using our cell line system, we spiked cholesterol into these cells and measured gene expression. So here we're just looking in the Wolbachia uninfected background and looking at TLR4 and the IMD gene. And when we add cholesterol, when Wolbachia is not in the system, we don't see any change in gene expression. When in a Wolbachia infected background, we don't see any change between the control and the cholesterol treatment for IMD. But for TLR4, we're seeing that cholesterol suppresses TLR4, and that only occurs in Wolbachia infected cells. It doesn't occur when there's no Wolbachia in the cells. So here was evidence there's this interaction between cholesterol and toll signaling. We did the reciprocal experiment, if you like, in that we used a molecule which would sequester biologically active cholesterol from the media. And so when we add this molecule and sequester cholesterol, we see an upregulation in TLR4, so the opposite effect in a Wolbachia infected cell compared to when there's the cholesterol freely available. So to us, this was suggesting that it's not a resource composition per se, and that cholesterol is a immunomodulating molecule, and this immunomodulation occurs specifically in Wolbachia infected cells, which is quite unique. So again, to further investigate this, we used our RNAi-based system and looked at knocking out these various genes under various regimes of cholesterol. So here's our control, and all these cells are infected with Wolbachia. And similar to our previous experiment, we see that Yang virus titer increases in a Wolbachia-infected cell as we increase the concentration of cholesterol. When we knock out the IMD pathway, we see no changes in viral titer. When we knock out ML1, which is in the toll pathway, we see that Wolbachia now can't protect. And similarly, we see the same with TLR4. Now, the next one is cactus, and I think this is the important column in the figure, if you like, in that now when we turn the toll pathway back on by silencing cactus, we see that now Wolbachia can protect again against, and this occurs even at high titers of cholesterol. And so cholesterol is turning off toll, but we're turning it back on by RNAi knocking out cactus. And so what I think we've done here is shown that there's a functional link between the two theories about how Wolbachia, or the two potential hypotheses about how Wolbachia can interfere with pathogens, and that at least in our case it appears that there's no resource competition, although we could investigate this further, but that cholesterol is interacting with the immune pathway. Now, how widespread that is in other vector systems, we don't know yet, but that would be something that would be interesting to investigate. Okay, so now I want to switch gears away from pathogens and talk about how Wolbachia is vertically transmitted and how this led us to investigating the microbiome. So one of the original goals of this work was to establish a stable infection of Wolbachia in Anopheles mosquitoes. So, and we were using this adult injection model. Now, we had multiple strains of Wolbachia and multiple mosquito species, and so we wanted to investigate what was the best combination that we should pursue this work with. And so we did this experiment here where we injected the various strains into the two mosquito backgrounds and measured ovarian titer, as well as the titer of Wolbachia in the ovary. In the carcass, sorry. And the reason that we're measuring ovarian titer is that Wolbachia needs to invade the ovary to be vertically transmitted to the next generation. And so we thought this was a good proxy for vertical transmission. What we see is the Wolbachia, the WALB strain, so the Wolbachia from Aedes albopictus mosquitoes, infects both the Anopheles species that we're working with better than Wolbachia from the fly. And that's probably not too unexpected. We're also seeing that the WALB Anopheles gambiae combination appears to have the highest Wolbachia titer in the ovary. So we focused our experiments on this combination. We also looked at how Wolbachia, or the titer of Wolbachia in the ovary over time, and we see that the longer post-injection, the higher titer of Wolbachia. And so this could be because Wolbachia has more time to invade the ovary, it could be Wolbachia is replicating in the ovary, or it could be a combination of both. We did some microscopy, and hopefully you can see, maybe if you look at the TV it might be a bit better, but we looked at the location of Wolbachia in the ovary, and we do see a heavy infection in the follicular epithelium, and these could be bacteria that are trying to invade. But we did see Wolbachia within the nurse cells of the oocyte, which would then be dumped into the developing ovary. So we were confident that Wolbachia was in the right location to be vertically transmitted. So from these experiments, our take home from this was that we needed to inject a high titer of Wolbachia into the mosquito. We needed to leave it as long as possible post-injection to give Wolbachia the opportunity to invade the ovary so it could be vertically transmitted. So we did these experiments. I injected mosquitoes, gave them a blood meal after 20 days post-injection, and then came in the next day to put my egg cup in the cage, and all the mosquitoes were dead. So that was a little bit disheartening. So we were wondering why this was happening, basically. And so there's a lot of processes that occur when a mosquito takes a blood meal, a lot of physiological changes in the mosquito. But one thing that we also knew is that the microbiome proliferated dramatically. And these microbes can produce toxins like ROS, which can affect survival. So we were wondering if there was were we upregulating or the abundance of microbes, and was this responsible for causing the mortality? So to address that question, what we did was we reared a line of mosquitoes continuously on antibiotics. And so we chose a cocktail of antibiotics, and these antibiotics did not affect Wolbachia replication. So we repeated the experiment again. And so we had a line that was reared conventionally on sugar and a line with sugar plus the cocktail of antibiotics. After repeating the experiment, we didn't inject a higher titer, and we blood fed them a little bit earlier, and it was enough. We did see mortality, but some of the mosquitoes laid eggs, and some of those eggs hatched and became adults. And so we assessed the progeny then for Wolbachia, and we were really excited to find that Wolbachia was indeed vertically transmitted. So this is the first time it had been reported, so it was a really exciting day in the lab. If we look at our control line here, the conventionally reared line, 10% of the mosquitoes were infected, and they were infected at quite low titers. When we compared that to the antibiotic treated line, we saw perfect maternal transmission. 100% of the mosquitoes that made it to adulthood of the progeny were infected, and they were infected at titers 100 to 1,000-fold higher. So this was indeed suggesting that there was some antagonism between the microbiome and Wolbachia for vertical transmission. We repeated these experiments in Anopheles Stevens eye mosquitoes, and we saw similar results in that we got vertical transmission when the mosquitoes were reared on this cocktail of antibiotics. NP stands for no progeny. Despite injecting 5,000 mosquitoes, none of those mosquitoes survived or laid eggs, and those eggs survived to get a progeny. So the fitness costs of Wolbachia infection in the mosquito was so severe. So we knew that our antibiotic regime was enabling vertical transmission of Wolbachia, and so we wanted to identify what bacteria we were modulating, we were changing by giving these mosquitoes antibiotics. And so we did next generation sequencing of 16S using a MySeq platform. And we compared mosquitoes with and without this antibiotic cocktail. And so the first thing that struck us when we looked at the data was that these mosquitoes that had antibiotics, they weren't sterile. They still had a similar number of reads. It was just that we were reshaping the composition of the microbiome. And a taxa that stuck out to us when we looked at these data that significantly changed in the antibiotic compared to the non-antibiotic mosquitoes was azaya. And so here it's quite abundant, and when we treat with antibiotics we almost remove it. Now, we knew from the literature azaya was a bacterium that infected the germline of mosquitoes, and it was in the gut, but also in the ovaries and in the testes, and it's vertically transmitted, both maternally and paternally. So it's occupying this sort of the same ecological niche, if you like, as Wolbachia. So we thought that there could be an opportunity for antagonism between these two species. So to functionally address that, what we did was we developed a third line of mosquitoes. We made an antibiotic resistant isolate of azaya, and then supplemented it back into this line. And here you can see the qPCR for azaya, looking at those three lines. And so firstly we have the conventionally reared mosquitoes that possess azaya. When we treat with antibiotics, we remove azaya. And then when we treat with antibiotics but supplement with the antibiotic resistant azaya, these mosquitoes all have azaya at similar levels to the control. So we were confident our notobiotic line, if you like, was what it should be. And so we knew that there were severe fitness costs on the mosquito after we inject with Wolbachia. So we injected Wolbachia and then just measured the mortality in these lines. And so when the mosquitoes feed on sugar, there was a small but significant decrease in mosquito survival in the conventionally reared lines. But that was nothing in comparison to when those mosquitoes were given a blood meal. And so here, after 2 or 3 days, you've got maybe 10 to 15% survival in this line that was conventionally reared. So when these mosquitoes are infected with Wolbachia and they take a blood meal, you see this high mortality. And this was the reason why we couldn't get progeny from the conventionally reared lines in some of these experiments. When we resupplemented with this antibiotic-resistant isolate of Isaiah, we see a significant change in mortality compared to the antibiotic treated control, but this Isaiah bacterium is not responsible for this mortality, and so it's another bacterium, and we think we know what that is, but I can talk about that later. So we knew that there was this blood meal mortality phenotype related to the microbiome, but then what we wanted to do was then identify if Isaiah was responsible for blocking Wolbachia vertical transmission. And so we're looking at these two lines, which were injected with Wolbachia and treated with antibiotics. The difference is one has Isaiah, the antibiotic-resistant Isaiah, and one doesn't. And so when we resupplement Isaiah into the mosquito, we block vertical transmission of Wolbachia compared to the mosquitoes that don't have Isaiah, and they're transmitting Wolbachia to their progeny. So here I think we demonstrated that Isaiah is responsible, and it's possibly one of the bacteria that can interfere with vertical transmission of Wolbachia. And so these data might go some way into explaining why Wolbachia doesn't infect Anopheles mosquitoes in the field, in that it's not that the vector is incompatible for the bacterium, there's this microbial incompatibility with Wolbachia. Okay, so let me move on now to some of my more recent work. And so our lab interests, if you can read that, I think they span across these various themes. And so that is of host microbe interactions, vector biology, and infectious disease. And I'm sort of sitting in here in the middle of these three disciplines. And so I think there's three main themes from my research lab. And the first is looking at the tripartite interactions between the microbiome, insects, and the pathogens they transmit. And so we know microbes can actually interfere with pathogens directly, so they can produce molecules which have anti-pathogenic properties. They can also influence the phenotype, many phenotypes in the insects, such as immunity, which could also influence vector dynamics and pathogen dynamics in the insect. Another theme is looking at the factors that shape the microbiome. And so we really have a poor understanding of what these factors are. We know that the environment is really important, and there's also that host genetics is important. And so there's some level of host control, but we really don't know too much in regards to that. One area that we're actively looking at is vertical transmission. And so I mentioned how Wolbachia is vertically transmitted as well as Isaiah, but apart from that, we know next to nothing about what other microbes are vertically transmitted, and so that's something that we're very interested in. And another theme is how we can engineer these microbes to either manipulate the mosquito and manipulate its genetics, or to manipulate the transcription of the insect. And so this could be useful as a lab tool, or we could deploy this into the field, and so we could modulate a phenotype. And because we're using RNAi, we can potentially modulate any different gene in the insect. And so I hope you can see that if we can combine some of these themes, we can develop strategies that could be useful for controlling mosquito-borne disease. So if we have a bacterium that blocks a pathogen and that's vertically transmitted, this would be something similar to a Wolbachia type strategy. Alternatively, if we can manipulate the microbe to change vector competence of the mosquito or kill the mosquito, we can develop strategies. And then if you couple that with the ability of this microbe to be vertically transmitted, this is something like a new type of paratransgenesis. Or we could combine all three together and have the microbe having an innate anti-pathogenic response and also engineering a second response in. And so you can have two mechanisms by which you interfere with the pathogen. Now, I'm looking at this in Aedes mosquitoes, and you're probably not too surprised to hear that I'm looking at Zika virus at the moment. So I mentioned the vectors Aedes, we're looking at the Zika pathogen. So what are the microbes that we're interested in? And so we've done some general characterization of the microbiome of Aedes aegypti and Aedes albopictus. And so here we have field caught samples that we caught with either a BG trap or a gravid trap. So they should change in their blood feeding status. And we have also lab samples as well. And so here we have the microbiome. Wolbachia has been computationally removed from Aedes albopictus because it dominated the microbiome. So we're looking here at other microbes, potentially gut microbes. What we see is Pseudomonas is a major component of the microbiome, and it appears to be infecting both these mosquito species. Serratia is dominant in Aedes albopictus, and this is a really common bacteria. So it's interesting that it appears to be completely absent from Aedes aegypti. And it's possible that this Escherichia shigella bacteria is replacing it, or so there could be some opportunity for interactions here. Again, here in the lab, the mosquitoes appear to be dominated by this enterobacteria and don't have this bacterium here. So I'm interested in these possible interactions here, which could also indicate some antagonism. The stars indicate that these bacteria can be cultured, which is important for any strategy. So we can actually work with these in the lab. And so here, this bacteria like Pseudomonas, it's in both mosquito species. So if we could harness this as a microbe, we could potentially develop strategies that are functional in both aegypti and also albopictus. Now, when you hear microbiome, I think you probably all think of bacterial microbiomes, but mosquitoes also have a fungal microbiome. And so we've done some sequencing of the fungal microbiome. They're dominated by this Malassezia, if I can say that properly, and also a Candida. So I think this Candida is interesting in that we have a lot of genetic tools to manipulate Candida, and so that could be something that could be useful for an applied approach. Now you'll notice here I've only got the albopictus fungome or microbiome up, and that's because we sequenced Aedes aegypti and we got 99% host reads. And so this is one of the challenges with working with fungal microbiomes, in that it's a eukaryotic microbe in a eukaryotic host, and they both have ITS. And so when we did this sequencing, we got 99% hosts. But we're developing a strategy to overcome this. And so here's basically your PCR. You've got the host, which is in greater abundance to the fungi, and we're developing this PNA blocker to specifically bind to the host, and that will block that PCR. And so we've done some work just characterizing this. And so here's some gels. When we add the PNA blocker, we see that we don't get this host band here. And so this appears to be specific because we've spiked the Aedes aegypti template with yeast, and so we get amplification here. We added this enzyme SPH1, which specifically cuts the fungal amplicon. So we're confident we're getting fungi here. We've also done this for albopictus and Culex quinx, and we've designed the probe, the PNA blocker, so it will interfere with ITS in albopictus, Culex, Aedes, and Anopheles. And so here we've got it working in these systems. We haven't yet tested Anopheles. So the next step is now to resequence those DNA isolates with this PNA blocker, and we're hopeful that we can get the microbiome of Aedes aegypti. Okay, so I mentioned they're the symbionts that we could potentially work with. I mentioned that one of the research themes was looking at the tripartite interaction. Now, most people that investigate this interaction do this with lab mosquitoes, and they can have a very different microbiome compared to their field counterparts. And so we're taking a different approach, and this is a Twitter-inspired collaboration in that I saw a tweet on Twitter that said that Alex Wild from UT Austin was catching 10 to 20 Aegypti per hour in his backyard. So I gave him a call, and then 2 days later we were up in Austin collecting for ourselves, and so we collected Aegypti, brought them back to the lab and fed them a Zika virus infected blood meal. And so what we've done is now looked at the infection in the mosquito, and we're going to correlate the microbial taxa in these mosquitoes to Zika infection. We also, the mosquitoes that didn't take a blood meal, we homogenised them and used that as an inoculum for plates, and so we have a range of different microbes from these mosquitoes. So the results of our field work were that we got 177 mosquitoes in our collection. We brought them back into the lab, and 135 of them took a blood meal. Of those 135, 112 survived for 10 days, which is when we assessed for virus, and 64 of them were infected with virus. So just over half. So we think we have a good breakdown of infected and uninfected mosquitoes to now do microbiome analysis and look for taxa that correlate to infection. Another theme was looking at vertical transmission, and so here we're isolating bacteria and transforming them with a plasmid that expresses red fluorescence as well as antimicrobial or antibiotic resistance, growing these up in liquid cultures and then spiking them onto insects. And so we did this, we spiked onto mosquitoes for 5 days. At 5 days, we then simply homogenized the mosquito and put that homogenate onto antibiotic plates. And so we can select for CFUs or these microbes that contain the plasmid. And so we could re-isolate both Enterobacteria and Serratia from these mosquitoes after 5 days. Interestingly, we didn't get E. coli, despite it being at the same time as we were infecting the mosquitoes. It's possible, we used DH5 alpha, and it's possible it's just a wimpy strain of E. coli, and it's really adapted to plates, and it can't handle being back into a gut environment. After 10 days, so the mosquitoes had been on clean sugar for 5 days. So here we repeated these isolations, and this time we dissected mosquitoes. So we had the carcass and we had ovaries, and we could detect bacteria in both of these tissues. And so it's likely that the bacteria are migrating from the gut and then then infecting the germline of the mosquito. The bacteria are fluorescent, so we did some microscopy on these mosquitoes, and we can see this is an image of the ovarian duct, and so we can see Serratia present in the ovarian duct of the mosquito. We gave these mosquitoes a blood meal and let them lay eggs, and then we cultured the oviposition water, so the water that was in the cup, and we could detect the bacteria in the water. Then we hatched the eggs and reared those eggs up, and what we found was that we could isolate only Serratia in the progeny of those mosquitoes. So what we think is happening, if you take this data together, is that Serratia is migrating and it's infecting the ovarian duct. And so when the mosquito is laying eggs, Serratia is being smeared on the surface of the eggs, and then those eggs are then infecting the larval water. So it's a type of vertical transmission, which is sort of a combination of vertical and horizontal transmission, I think. And I think this could have implications for strategies to release, and that you could spike larval water and mosquitoes can acquire the bacterium. Also that if a mosquito with your bacteria Serratia lays in a larval pool, other mosquitoes that are in that pool could also acquire the bacterium. So that might have some type of spread strategy. Okay, and the last theme I want to talk to you today about is how how we're manipulating bacteria to be a delivery vehicle, if you like, and we're interested in using bacteria to deliver RNAi to the mosquitoes. And so we're undertaking two approaches here. One's perhaps more complex than the other. So I'm just going to talk about the simple approach that we're developing at the moment. So we are expressing RNA hairpins from a plasmid, and we have RNA hairpins targeting this COP1 gene, which is a cotamer 1 protein. It's been shown, other work has shown that when you silence this gene and the mosquitoes take a blood meal, they die. So this was a really nice phenotype for us to assess, a quick and simple phenotype for us to assess if our delivery strategy is working. And then if so, we'd go on to knock out other genes which might have other important traits. And so we also have a bacteria expressing hairpin targeting GFP as our control. So we grow we're growing up these plasmids in E. coli, and then so what we can do is just spin down the bacteria and do a quick RNA extraction on the bacteria, and then transfect that RNA, which should contain the RNA hairpin, onto insect cells. And then so we've done that and done qPCR, and so what we can see is that we can get gene knockdown in using this system in cell lines. And so the next thing we did was feed these bacteria expressing RNAi to mosquitoes. And so we had our GFP hairpin versus our COP1 hairpin, and then we monitored the mortality of these mosquitoes. So when they take a blood meal, so when they're fed on sugar, there's no difference in mortality. When the mosquitoes take a blood meal, the mosquitoes that have the bacteria expressing the hairpin against COP1 have a high mortality rate. So we're confident this system is up and running. The next approach that we're going to use this system is to interfere with Zika vector competence in the mosquito. Here we're targeting the virus itself, so the NS5 or the NS4B gene of the virus, and we're also targeting the mosquito. So this is Pierce, which is the negative regulator of the JAK-STAT pathway, and this pathway is known to have antiviral properties. Again, when we do this, we grow up the bacteria, do an RNA extraction and transfect this onto cells, and then challenge those cells with Zika virus. We can see that the cell lines that have the RNAi or RNA hairpins have reduced Zika compared to the mosquito. So we're confident that our bacteria are producing these hairpins and that can interfere with Zika. And so we're really excited now to go and put these into mosquitoes and test for vector competence. So, in summary, I've shown that Wolbachia has potential for malaria control, but we really need to evaluate the other parasites. That Azaia can interfere with vertical transmission of Anopheles. Onyongyong virus, or Wolbachia can inhibit Onyongyong virus in the mosquito, and that seems to be mediated by the toll pathway, and cholesterol modulates that toll induction. We're investigating the tripartite interactions, and importantly we're doing this in field mosquitoes. We're looking at vertical transmission, and we think we're on our way to developing a strategy for bacterial delivery of RNAi to interfere with Zika. So with that, I'd like to acknowledge that most of the work on Wolbachia was done when I was a postdoc in the Rasgon lab, and that was at Penn State and Johns Hopkins. And the latter work that I described on the microbiome was done by two talented people, so Shiva, who's a postdoc in the lab, and Miguel, who's a PhD student in the lab, and our funding bodies. And with that, I can take any questions.
Ben.
Do you think the cholesterol story also applies just to regulating Wolbachia in the absence of any virus? Do you think the toll pathway stimulation could be affecting Wolbachia density? Have you got any evidence?
We haven't looked at density, so that would be interesting to do. It could be that the change, or Wolbachia in an uninfected line, we just don't have that high level of toll stimulation, and so if we artificially enhanced toll by RNAi or overexpressed it and then put cholesterol on, we might see a similar reduction. But it is interesting that there seems to be this interaction. But yeah, we haven't teased that apart any further.
If you look for sensitivity to challenge those in those studies, the viral challenge doses, so the concentration of the virus in the blood.
Yeah, we haven't titrated the virus or anything like that. That could be interesting to look at. Yeah.
Do you know what causes such a profound difference in different Wolbachia strains? It's my understanding that they're quite...
For pathogen interference? No. I mean, we're dealing with an A and a B group. But as far as why we have differential responses to Plasmodium, yeah, we're not sure. It could be a nice system to tease that apart. There's not a lot known about the different host responses to falciparum and Berghii, and so we could look at specific genes to say, is Wolbachia using this method to enhance, but that suppresses falciparum. There's one gene, and I can't remember it off the top of my head, but I know there's a gene that Wolbachia upregulates that's been shown to suppress falciparum and enhance Berghii, so that would be the first one I'd look at.
Do you have any explanation for why Anopheles only transmit one virus? Lucky?
Yeah, I don't know. Yeah, but that's a general question in arbovirology as well, for what makes a host a competent vector.
And with the enhancement, is that a consistent finding? It's always enhanced. It's never a dynamic, no change, or sometimes a reduction. It's always an enhancement, is it?
No, so there's variables can change that. So I didn't get to talk about that, but in our Plasmodium uellii work, we looked at temperature. And when at different temperatures, we see different effects. And so at temperatures optimal for uellii development, we see enhancement. I think when it was hotter, they were suppressed. And when it was colder, there was a different phenotype. So
does that link to the immune?
Yeah, we don't know. Possibly. But yeah, so it seems that temperature is changing or modulating the effect Wolbachia induces on the host and or the pathogen.
And then the next big question is really, are you concerned that this effect could exist within some of the viruses, which are...
So...
There hasn't been any evidence of an enhanced viral...
No, that's not true. So... West Nile virus, we've done some work in Culex, and again, so the caveat with our work is that we're injecting Wolbachia into the mosquito, so it's not how it's naturally acquired. In stable infections, there hasn't been any evidence of enhancement, but when we inject Culex tarsalis with Wolbachia, we see that they become better vectors for West Nile. I think there's some work coming out on Rift Valley fever that shows something similar too. But these aren't in 80s, and it's not in stable infections.
So you're not worried about it?
I wouldn't say I'm not worried, but I'm just saying that they're the caveats with our work.
Further questions? If not, that leaves me to thank Grant. Thank you very much for your presentation and the enormous diversity of work you're doing.
Okay, thanks.